Hello!
Hi there.
Welcome to Microbiology Journal Club, where we nerd out big about all things small.
My name is Danny, and in a previous life, I dropped out for my PhD in microbiology, where I was infecting skin I grew with MRSA. Used to be a fact checker for pharmaceutical advertisers, and nowadays I'm the president of Biotech Without Borders, a nonprofit based in NYC dedicated to aiming the public with the tools of biotech.
My name is Faz. I have a PhD in microbiology. I've mostly worked on bacteria, but I've also worked as a research integrity specialist and I'm currently working for a scientific journal.
Every week we meet to talk about microbiology, and today we're doing an overview of some of the coolest microbiology papers that we've seen during the week.
And at the end of the month, we'll be looking at microbial ink and doing a deep dive into how that paper works. And traditionally at the end of every month, we pick one paper and do a deep dive on it. So if you have any papers that you are interested in, please let us know, and we can add it to a list of things that we might want to do at the end of the month to sum up.
Yeah, you can see the list of the articles that we cover in this session or the potential suggestions that we're going to do deep dives on in our shared Zetaria library, linked in the doobly-doo below.
And we want to hear from you, so please use the comments or tweet us at microtwjc. And boy, do we have a show for you today. Firstly, monkey genitals. We've got lots of those. Well, we got three, but yeah. How is that related to SARS-CoV-2? Well, you have to tune in to find out. What else do we have? We have various things on, various molecular stuff to do with SARS-CoV-2. We've got like the loss of smell or taste, what genes might be involved in that. Very interesting 23andMe study. Let's see, what else have we got? We've got mucins, mucus. How does your mucus inhibit SARS-CoV-2? And what does that mean for transmission from fomites? And what else are the cool things? Because I know we have some.
We got spillover events from deer.
Yeah. We've got like injecting stuff into fungal cells, puncturing them, almost like with the vampire-like teeth. That's something that I found quite fun. Also, like artificial organelles for bacteria, and also a way to make CRISPR much better than it actually is. So, well, I guess let's get to it. So we've got like our first paper, which is to do with SARS-CoV-2.
Antibody escape and global spread of SARS-CoV-2 lineage A27. And so this story is from the beginning of 2021, like end of 2020 into 2021. So it's not, this is like classic, like scientists looking back at something and trying to learn what it tells us. And specifically during this time, they're looking at viral surveillance in Germany, I believe, and they find this lineage A27. Did you hear about it on the news? I didn't really hear about this.
I think there's a period of news where people were worried about every single variant that they found.
Yeah.
And so I remember there was AY3. I think we did a show on that. I think we missed the boat on A27 because it's. I think like there's a lot of things.
There weren't that many infections with it, right? Like I think that's maybe one of the reasons why we might not have picked it up initially during that news. But what's interesting about this particular variant is that it has these immune evasive mutations, but it comes from a radically different lineage than almost every SARS-CoV-2 strain that we have in circulation now. And we've spoken about this mutation, the D614 mutation, where that original strain in Wuhan didn't have it, the original in China, originally there wasn't that mutation, but very soon afterwards it took over. And the way we thought about it is that like mostly it was that strain with the D614G mutation that ended up like spreading globally. But as with most things in biology, there's always small numbers of very small amounts of variation that can go on to give rise to new variation that then we detect later. And so the story here would be that something about that very first ancestral strain before it picked up the D614G mutation, which we've seen much data now to suggest that it really does improve fitness quite a bit. Without having that mutation, that has been in circulation in certain populations, and during the time that it's been in circulation, it's picked up some of the same immune-evasive mutations that we've seen in some of the scarier, more prominent variants in the world.
Oh wow! So essentially, it's related to the original version of SARS that was first found, but it didn't pick up the D614G thing. So it's just been hiding out. So it hid out for effectively like a year without anyone noticing it.
Yeah,
and
probably like low
popped up in
levels of
2021.
infection. Yeah, exactly. Low levels of infection. And then I guess some surveillance. Maybe surveillance had always seen it, but the reason why the researchers are drawing attention to it in this paper is just to show people that idea that convergent evolution happens, right? I mean, this is like an everyday sort of thing. Like if there's pressures, if viruses are left given time, right, to sort of explore their mutational space, they're going to find similar solutions. Even if they came from radically different lineages, like, you know, this A27, like, it's not very good actually at replicating in things. They stick it in some models later and they say it's terrible at replicating, right?
Still good.
Yeah, it doesn't have some of the mutations that are required, you know, I guess that were in the successful lineages that allow them to just be good replicators. But, you know, there's still a pressure to evade the immune system. And so they picked up those mutations along the way, just with the low level of infections that they were able to sustain in the world.
Yeah, it's nice to hear about a variant that I don't have to be afraid of. It's nice to hear about a variant that is just like, oh, this one's just pretty bad actually, but we can still learn some interesting things from it.
Yeah, it's like more of, it's like a side note variant in many ways. Like we don't expect, like, I don't think the researchers expect that this is going to be the next big thing that's like going everywhere. Rather, it's being able to sustain some low level of transmission over the course of who knows how long. And again, it's picking up things that make it more immune evasive. So I guess that piece of evidence is sort of telling us that, you know, the virus does want to go down in that direction, right? We have our own, the immune system does a good job of suppressing viruses, but, you know, that's not, viruses have many tricks in their toolbox, right? I mean, these are the tricks. Mutations are the ways in which they find the gaps in order to continue transmission.
So yeah, this is like a cover band like variant where it's like they can play some hits well, but not all of them, and we can figure out which ones are actually important based on which ones they can hit. Because you can have a Beatles cover band that does "I Am the Walrus" really well, but if you can't do like the good songs, then it's just not gonna work.
Yeah. I think that's a nice little analogy that we could apply to this. So yeah, the reason they know that it has immune evasiveness because they do the classic neutralization assays. They know that it's not a great virus in general because they infect cell culture. I think they do mice too. Yeah, they do cell culture. Or I'm not sure. They at least do. Oh yeah, they do human ACE2 mice as well.
Yeah.
So yeah, that's the report of A27. Again, good to know, right? Like I think these stories help us get a grasp of, like, it can help inform like what the long-term direction is. Mutation doesn't stop for anything. It's plugging away, trying to find a way to be more successful. Hopefully that doesn't include giving us more severe disease, but you know we have the vaccines for that particular element.
Right. Up next, we've got highly divergent white-tailed deer SARS-CoV-2 with the potential deer-to-human transmission. So this is an interesting, because I think like we've covered a little bit this past year over, like, we know that white-tailed deer do get infected with SARS-CoV-2. There's been a jump from human to deer in those populations, and this has been essentially like they've did some monitoring of deer where they went out and well, also the thing is it's hunting season, I think, in North America. So based on that, they were able to sample some of the hunted deer to see whether they sampled the hunted deer to look at like the strains of SARS-CoV-2 that are evolving in the deer and seeing what's happening with those. Again, this is kind of on a similar theme of like SARS-CoV-2 evolution in strains that aren't necessarily things that we should be too frightened of, but we can still learn something about.
Yeah, I mean, good to keep our eye on it, right? Because these are the places where we may see emergent variants. Not now, but like perhaps like in the future. And I will say, like, you know, reading this story, it also reminded me of like how we talked about Ebola a little while back, right? And how it always comes from a spill, or well, always until 2021 and that one event, but it mostly comes from these like spillover events. So, understanding like spillover events from deer is like sort of the entryway to understanding like, are deer going to be like our new like watch out sort of moment, like whatever the initial evolution that happened that let this emerge out of China. Now it's in human populations, but are we seeding it into new animal populations that will then be things to sort of monitor more carefully?
Yeah, I do feel that's why they're so intent on monitoring the evolution in these deer. And I think they found that these deer variants are turning out to be quite different from the other variants. So they've got this like big like cladogram, which there's a graph of showing the family tree and how related they are. And the variants they found in Ontario, which from deer, seem to be quite different from the other strains. They are evolved from bee, the bee cluster. So, but the question is, whether they can be dangerous. And they did find like one or two people who had, so I think they looked around like sequence records to see whether there are people out there who had similar strains, who were infected with similar strains to what was found in deer. And they found, I think, one person who was themselves like, I think, a deer hunter, or I don't want to say deer involved person because that might have some connotations, but
deer enthusiast.
But
no, I mean, that's also kind of strange.
A deer enthusiast. I mean, that's one way to describe someone who hunts deer.
This would be the first then report of something like this, right? Like I think we've seen in mink, right, that there were definite signatures of transmission between first humans to mink, but then mink back into humans, and so that's what sort of that instigated the large culls of mink that we saw globally, in particular in Denmark, which they first identified this.
And of course, more recently in Hong Kong with hamsters as well. Very similar thing happened.
Yeah, we didn't cover that news, but I saw it in the introduction here of this paper. I didn't realize that pet hamsters, which makes sense. We've been talking so much about the hamster model of disease. Like pet hamsters are really just a step away from that.
Yeah, and another thing that they did, they wanted to make sure that see whether this strain was more immune evasive or had some. And they found that, no, this strain also reacts to vaccines in the same way as other strains, which is which is good news. So it means that neutralization is still at the same levels. And yeah, we don't have to worry about this. And also, it brings back to the fact that even though this strain is very different, it's very kind of divergent from the other strains, it still gets covered in the vaccine envelope that having a vaccine based on the original strain covers all the strains, which is it kind of speaks to the necessity to keep the wild type in the vaccines at the moment.
Oh, for sure. Yeah, you don't want to over specialize vaccines or boosters in a sense. Yeah, I mean, it's I don't dear. I wish that it wasn't the case that there was so much human spread that it got into all these populations, because again, the best medicine would have been just prevention in the first place, right? Not as many human things, so don't get into the deer. But it's nice that there are scientists out there watching these things, and hopefully it encourages research along these lines, because I think viral surveillance is a really important part of global health. That's one of the things that we've been taught the hard way in this, because just viral surveillance programs can be incredibly useful in wildlife, right? In zoonotic reservoirs where we might see crossover.
I completely agree. I think they're so important, and there are lots of labs out there that specialize in virological surveillance. They go to areas of where there are likely spillover events and try to catch viruses before or when they spill over. I mean, I think there's a reason why quite a lot of viruses are named after areas in Tanzania and the West Nile, because that had a really impressive research institution that meant that lots of viruses are named after landmarks in certain countries, and it's kind of interesting to be a tourist in those areas and go, oh, I'm going to swim in the Ebola River, and
and I wanted to say one more thing. It's eluded me though. It's something about sequencing centers. Okay, never mind. Let's move on. It might come back to me.
Speaking of sequencing centers, 23andMe has some.
Oh yes.
Yeah. So this study comes courtesy of 23andMe.
Yeah, a good old GWAS, the most correlative of data sets that you can do, essentially leveraging the power of huge amounts of sequencing data and linking that to phenotypes, finding that statistical needle in a haystack to say that these phenotypes are likely associated with particular genotypes. And we know as 23andMe, their database, it's SNPs, right? So they have all these single nucleotide polymorphisms in a whole bunch of different genes that allows them to, deliver sequencing, or I guess it's not particularly sequencing per se, but it allows them to deliver this or analyze this data set across a huge number of individuals. And they have the loss of taste or smell as one of the questionnaire questions that got delivered. And so they said, well, let's find the genetic associations with that particular survey question. So, limitations inherent in that are it's a survey question. So that's like self-reporting bias and all these types of things. But they do pull out a very strong signal for something that seems relevant too, like, at least theoretically, this UGT2A or UGT2A2 locus, which is some sort of lipid. It participates in some sort of metabolism in the nasal epithelium.
Yeah, so I think during olfaction, I think these enzymes are expressed in the olfactory epithelium, and they're involved in the elimination of odorants that enter the nasal cavity so that you don't kind of smell things for too long. And I think that they're kind of based in the processing of like smell molecules. And I think what they say is that, okay, well, we kind of know that the olfactory epithelium, the epithelium inside your nose, gets damaged during COVID infection, and that which can completely inhibit the sense of smell. And that these two operons people who like lose their sense of smell are more likely to have specific mutations in these two like kind of areas of the genome.
Yeah, so like it could be like they're trying to say this might be a susceptibility locus, right, to the disease phenotype. That like if you had these if you have these mutations, then you may be more susceptible to losing your sense of smell.
Yeah, I mean, I think like the counterfactual as well
upon
is
COVID
just
infection.
as interesting, where certain of these variants make you more resistant to the loss of sense of smell caused by SARS-CoV-2, and the question is like because like SARS-CoV-2 is causing damage to the olfactory epithelium, so the question is what makes it still function even in that situation. So that's, but yeah, you can look at either way, but I think it's quite an interesting avenue of investigation they've got there.
Yeah. Again, GWAS is usually, I'm not sure if we've covered that much GWAS. Like this plot that you have in front of us is a classic GWAS plot, the Manhattan plot, right? where they've done like a statistical comparison, like does my gene of interest associate with this particular phenotype, and they assign like a strength of that association, statistical strength of that association across all the different loci that they look at. And so the idea is that if there is some sort of association, it's not going to be in every individual that they test, right? But like they'll see some of the samples like rise up above the noise, and you get like a skyscraper, if you will.
Yeah.
And then you can look at the tops of those things, and those are your candidates for association.
Yeah, I think there's some interesting thing with this data, because what they did was they segregated it into separate pools, I think based on race or like kind of genotype, and then they tested each individual pool, and they did a meta-analysis overall to see which kind of functions still held. So I think that's quite a good.
Yeah, so they're pulling out all their statistical rigor, right, in order to make sure that they're not creating spiralist correlations that don't have a strong backing or could be explained by some other variance in the data.
Yeah, so I think that they definitely tried their best, but even they know that this isn't the perfect cohort, but it's a great way to generate a hypothesis that other people can test later.
Yeah, so like the idea would be that now you can, make this mutation in a mouse or the hamster model maybe, right? Run that through an infection, and you get a better understanding of, you might potentially find some very important thing about, this particular disease process within SARS-CoV-2.
Yeah, hypothesis generating research is kind of an important part, even though like you get a lot of like bad hits from it sometimes. It is still like very important and can be quite useful. But yeah.
Okay, that's all I want to say about that.
Yeah, and the next paper is a preprint called An ImmunoPET Probe to SARS-CoV-2 Reveals Early Infection of the Male Genital Tract in Rhesus Macaques. So this is
monkey junk.
Yeah, this is a paper about monkey junk. So that's the thing. That's what initially they weren't looking to study that. I mean, this is something that so this is used as...
You want to talk about maybe immunoPET?
Yeah, immunoPET. So positron emission tomography is a technique based on giving a radioactive probe to an animal, and that radioactive probe produces a positron, and that positron runs around through like space until it hits anything, and when it hits, it will emit a gamma ray, and that gamma ray can get detected by a detector that you have around the animal. So
it's a very sensitive method to find small amounts of anything. You have to tag, right? Like the idea is that it has to work with the immuno. I guess part of it is saying that the PET probe, the thing that emits these positrons, it's being linked to an antibody of some sort. So then that gives you the ability to essentially anywhere an antibody can go and bind to something, right? You can get this. You can get a signal out. And that gamma ray, as you say, I didn't know it was a gamma ray, but like whatever, that energy that emerges from the probe, like that goes through everything. So you can image very deeply into tissues.
Yeah, and you can produce some really snazzy 3D images like these ones here, which I need to get the animation working.
Sure. So I'll just continue as you do that. So immunoPET is this very great way of doing finding a particular antigen, and what's going on inside of a large organism or a large sample of some sort. And so they use it for SARS-CoV-2 in the rhesus macaque model. So the idea being that rhesus macaques, they're more similar to us. We've said it many times, right? It's like that closest animal model that we have. They can infect these macaques with the intranasal infection route, and then they dose them. So they let that infection go for however long. They dose them with this antibody against SARS-CoV-2, and they just infuse that. They infuse their whole body with this particular antibody. And if there's SARS-CoV-2 replicating in specific zones, the antibody will bind to it, and that will localize the PET probe to that specific region, concentrating it there. And so those regions will light up on the imaging software. And so it's a great way to go in and just say, where is SARS-CoV-2 going in the body?
Yeah.
And that's something that we know it's not just the lungs, right? Like we've seen so many studies now of all the different places it goes. This approach that the researchers use is just a really great way of getting at that, right? Like asking that unbiased question, where does it go? And yeah, so they didn't set out to study the male genital tract of rhesus macaques, but boy, did they find such a strong signal there.
I mean,
that's what it became.
I have a lot of sympathy for them, because this was basically very similar to what I did in my PhD, and I found like in female mice, their genital tract got infected as well. So I have some sympathy with them. I mean, here you can see where you expect the SARS-CoV-2 to be. So the nose, the lung, you can see like very nice images of the lower lung. Also, another thing to note is that this imaging shows the presence of the antibody rather than the SARS-CoV-2. So you can see there's this giant signal coming from the kidneys. That's because like you're looking at where the antibody collects as well during the infection. So at the moment we're kind of ignoring the kidneys for the moment. Another thing to point out is that they also take out the organs individually, and the lungs are much brighter on this because it depends on the density of the tissue. Because in order for positrons to emit the gamma radiation, they need to hit something. So in any place with lots of air in them, they don't hit anything very quickly. So that's why when you take the lungs out and squish them down, they produce a much brighter signal. So So
yeah, because they did the whole body of the rhesus macaque, but they also did a necropsy afterwards, right, and take out all the organs. Yeah, so that's where you get the squished
yeah, I
ones
think all the
from.
organs are a bit more squished, so you do get a much higher signal. That's just part of the imaging techniques. We also have another image where they just focus in on the lung, just to look at where the SARS-CoV-2 kind of goes. And but of course, the thing that we all want to focus on is what happened next, because what they noticed was that uh, that out of nowhere the male genitals were getting infected with SARS-CoV-2, and this kind of like we've had some hints about this in previous research, right? Because the ACE2 is expressed in certain cells in the genitals. And I think there were a few studies of like SARS-CoV-2 being detected in sperm or something like that. But yeah, this is kind of very, very obvious evidence that SARS-CoV-2 can move to the genitals for quite early infection, actually.
Yeah. So, I mean, it would be good to know if they were human-based, like, monitoring studies for this type of thing. I'm thinking about, like, I remember we also looked at one paper where they went through, like, different tissues, human autopsies, right? And they were looking for SARS-CoV-2 signals in the histology. I don't think they ever looked, actually, at the genitalia in that study. So, right, like this is one of those things where it's like you're only gonna find things that you're looking for, and having a paper like this, have this report in an animal model, like will help direct the attention now for folks maybe if they have those frozen samples, biopsies like still frozen down in the freezer, time to get them out, slice them up again, and stain to try to see if that signal is also present in some of the COVID-infected human individuals.
Yeah. Yeah, we've got like various days. I think they test out Delta and Alpha variant as well to see whether there's a difference. And this study was done, I think, on only 3 macaques. So we're not going to get any definitive idea of what differences are. But I think they've seen it in more than one monkey, which I think is enough for them to start thinking that, okay, this is something that maybe we should look at. And yeah, so I think is more just like on that area. And yeah, so it seems like there's more signal coming from
really
there.
See that, yeah, the outline of these genitals, right? It's so clear. There's like so many pictures of monkey balls here in this paper.
Yes, we're this is very much a paper that is, I think, both it's very informative and very exciting, but it also like kind of tips into the thing that something that people are quite uncomfortable talking about, which is it's interesting to see how people are reacting to it.
It's scratching my childish urge to be like, look at this, like scientists just staring at monkey balls all day.
Yeah, I mean, I haven't made the obvious macaque pun, and I do want extra brownie points for that. So please like in the comments for me not making that pun.
But yeah, I'll also say what I find fascinating here is I'm pretty sure like people use this PET scan like there's a lot of use in the medical field, right? Like when people are trying to diagnose like sort of rare cancers and things, like they want like a really sensitive method to look at every tissue and see like where there might be like, you know, potential tumor cells or whatever. It's like really great, I guess, to see it applied inside of this research context and just to illuminate like that's there is an interplay sometimes between the technologies that we end up using for therapeutics and the ones that we might end up using for research.
Yeah, I agree, and I think like for imaging technologies, there has been like an improvement over the past few years. Because I remember like back a couple years ago, I was working on imaging bacteria in like a similar way using bioluminescence instead, and I know that at the time there was like a lot of talk about creating like animal level, because a lot of the technology was designed for humans, and I think they've like, and the immunoPET probe technology is another like kind of great advance. And so it's great. Well, the great things about this is that these sort of technologies allow the bacteria to tell you, or the virus or the microbe to tell you where it is, rather than a lot of like the studies we have are like people going out and looking, whereas here like I mean, I think we've said a couple of times that where you find SARS depends on what tissue you sample, but for this kind of study, you are effectively sampling everything with the imaging technology, so it allows SARS to tell you where it is. So I think it's quite beneficial in that kind of respect.
Yeah, absolutely. And you don't have to, what's fascinating to me as well is like this is the intranasal route of administration, and yet you see it go everywhere, right? So like it just like it triggers automatically seeing this data, like all of those questions, which I mean we have been asking already because people have done the fishing experiments in different tissues, but like how does it get there, right? Like why does it replicate in those spaces? Like all of those hypotheses flow from seeing this data.
Yeah, and I remember like one of the things that I kept getting asked, because when I was seeing genitals get infected, it's like, we're like, oh wait, do they like scratch themselves down there as well, or like is there kind of? I mean, like, but that's different because I was working on the wound infecting microorganism as well, so the idea that grooming behavior could spread it. But this is this is fascinating. Enough talking about myself. I need to talk about the paper. Sorry.
No, it's good. It's good to see the relevance, right? Like because like I think that's an important thing to remember. Like it really matters. Like the circumstances in which the infections or the pathogenic microbes that we're studying, like the circumstances of those infections, like because they bring in concerns about how the model is being used, right? And valid criticisms to like, why did you see it happen in a certain way? In this one, I mean, one thing that comes to my mind automatically is there a sexually transmitted like form of this? Since like there's like so much replication in these tissues.
I mean, that is quite, because I think that some people are concerned about COVID-related infertility, and I've seen this paper being sent around as like kind of advice for men to please get vaccinated or else this might happen to you. And I think there might be some papers about that, but I'm not to
I
not
have no
enough is
idea.
out there yet.
Right.
As if like there weren't already enough reasons to get vaccinated. I mean, like you can't have sex after you die. I mean, people do know that, right?
Yeah. Like. All right, let's move on.
Okay. All right. So next one is Mucins inhibit coronavirus infection in a glycan-dependent manner. So this is SARS-CoV-2 has a spike protein that has all these sugar like kind of molecules on it called glycans, and they kind of play a role in, I think, attaching to cells. And mucins are these kind of proteins that are also covered very much in like glycans and this molecule called sialic acid that help to defend like the mucus, I think, against certain viruses. So any virus that has to live in the mucus for any amount of time has to be able to deal with mucins. So like with influenza, they have like sialic acid binding motifs, and I think like coronavirus also has a way to bind sialic acid, even though it isn't involved in the actual infection. It just is able to bind it, which is both a kind of a good thing and a bad thing. And this paper delves into what exactly that means. So
so this is about the spike protein on the surface of SARS-CoV-2 virions and its affinity towards these sialic acid residues on host-derived, right, polysaccharide. Essentially, it's a big component of mucus, right? It's snot.
And mucus is everywhere, and where we find that. And what they did here was they took some of this mucin and they mixed it in with SARS-CoV-2 to see whether it like helped infection. And also they created other versions of the mucin which don't have the sialic acid. So they had an enzyme chew off the sialic acid and use that as a control. And then the important thing they did here was also test it at different water concentrations, because all these interactions require lots of water. But when you like spread out in airborne droplets, I think we did a paper a week or two ago that was talking about how the air dries out those droplets, and that can affect how much coronavirus survives in it.
I mean, even the drying out effect, it like creates crystals of salts and things, right? And those things may not be incredibly hospitable to the virus. So I think one of the hypotheses they were trying to directly address in this paper is that idea that is the effect from binding the sialic acid, or is it from like having water be associated with these mucins, right, that create like a more hydrated environment for the virus?
And I think what they found was that having as things dried out, the mucins kind of coated the virus, and that plays a role in the virus not being able to cause infections. So the idea is that if you like sneeze or like gob out coronavirus like saliva onto something, it's not going to survive on that so long because the drying effect means that the mucins are going to become much more active against coronavirus. So
yeah, I mean, just in some way, this is just tooting the horn of our evolution, right? And actually, I guess all mammalian evolution, because it sounds like this is in cows and right. Mucins are all over the place in many different species of mucus. But you know, there's a long history in the development of that molecule and its widespread use within certain body plans. And one of the reasons could be it has this like multifunctional ability to prevent a wider range of viruses, things that seek to bind, that can bind sialic acid, I guess. I imagine that this has like a lot of relevance for actual viruses that use sialic acid, right, and these particular sugar moieties in order to gain entry. So this is probably a very wide functioning phenomena that we're seeing. It just so happens right now that it's being followed up on in SARS-CoV-2.
Yeah, and I think like for flu, they might have a very different like aspect of that because flu apparently does use mucins to kind of survive, but whereas SARS-CoV-2 isn't built like that, so that it's built differently. So yeah, I think that's all I need to say about that paper.
Cool.
Next one is synthetic virions reveal fatty acid coupled adaptive immunogenicity of SARS-CoV-2 spike protein. So this comes from like some structural findings that again, this is going back to spike protein because spike protein is really important to this. So the idea is that spike protein has like certain conformations can be open or closed when it wants to bind something, right? So it's got a receptor binding domain. And what they found during structural is that there's this little like kind of lipid tube that goes through it. So there's a tube that is almost like that can find lipids essentially. So like lipids are like kind of long chain fatty acid molecules that can slot into there. And when they slot into there, it causes the structure of it to close, which means that it's less able to bind. So this entire study is trying to find out about that interaction. So synthetic virions are kind of something they created on the side to investigate this. So they took a bunch of spike protein, put it onto the lipids, and created like kind of something that's similar to like SARS-CoV-2, and then like kind of popped out some very small like lipid bubbles that look very similar to SARS-CoV-2, and they call that.
Yeah. Well, I mean, look similar in the sense that they have spike protein, but they have nothing else. They're very minimal, actually. Yeah. I would say they're probably nothing like SARS-CoV-2, except for the fact that they have spike protein on the outside.
Yeah, that's right. And I think that's the point. The idea there is like selling that model and saying, okay, this is just a safe way to study SARS.
Yeah, they say it's a BSL-1, thing, because it essentially is just protein lipid mixture. It's not like a pseudotype virus where it's still a virus underneath there. It just happens not to be the particular genetic payload that you're looking at.
Yeah.
It's not even a single replicating form, right? Where you could imagine that like maybe something goes wrong or like some other piece of that DNA or some other piece of that RNA ends up being used in a different process. Like there's nothing here that could self-replicate.
Yeah, I mean, so this is quite safe and quite, because it's safe means that you can do more with it. You don't have to talk to various like health and safety organizations, or I don't know what's the US, but like talking to the government and saying, hey, we're not making this a terrifying bioweapon. Like it's very easy to work with.
Right. And of course, this technology probably could be extended to study a whole variety of things, right? Like insert your favorite membrane-bound receptor here, right? You can create small lipid particles that can, you can sort of see the binding of those receptors in this realistic in situ or in vitro style like experimental setup.
So this is all to study like the spike protein and to understand what this kind of lipid binding domain does. And what they find is that it affects. So there are certain types of fatty acid that cause it to shunt, and then they kind of speculate that there is a adaptive function to that. Because during the immune response, there are various like steroids that are produced by the immune system that are like fatty acid molecules that are used as signaling. So the idea is that when those are produced, that means that causes the spike receptor to close, and that means that the receptor binding domain, which is very immunogenic, that hides it from antibodies, it hides it from the immune system, and makes it more so. It improves survivability, but also because it makes it close, it also makes it worse at infecting. And so they also tested out some drugs and found like that dexamethasone can activate this fatty acid binding pocket, and because of that, it can stop SARS-CoV-2 from being as infectious. So I think this is like kind of got a nice mix of understanding virus and also finding ways to counteract the virus at the same time.
Yeah, totally. I mean, through understanding, this has always been, I think, as people who've studied infectious disease, that is the reason why, right? Like, you want to study infectious disease sometimes just like from the basic biology side of it, because those things will end up informing therapeutics later on, may end up. And in this case, yeah, that's one of the things that they're picking up.
I mean, it's quite interesting because like dexamethasone was one of the first like big like drugs that came out when we were studying, when it was the first one that was shown to have like a protective effect in SARS-CoV-2 cases. So it's quite interesting to find this other kind of functionality for it.
Right. Yeah, it could have been doing, this is underpinning mechanism, right, for that success that we saw on the clinical side.
Yeah. So
cool.
Yeah, cool. You've got like one last paper on COVID-19.
One last paper. This is a big one and a strange one too. I hadn't really heard about this. Vaccine-induced COVID-19 mimicry syndrome. And so in this paper, they're following up on one of the adverse events from the DNA base, the Oxford AstraZeneca vaccine. That in all the adverse events that they logged, this one had a slightly higher one for like a very rare thrombosis that. Right, I think we've heard about this, right? Like that's why like there was some increased monitoring, right? And they're trying to say like, why does this one have it, but not? Why didn't we see that inside of the mRNA style vaccines? And what they propose as the reason for that is that format factor, right? It's DNA versus RNA, and specifically in DNA, the body. Like the human cells, we use DNA and we splice DNA. But when we create like a synthetic construct, is just the DNA version of this RNA virus, we don't take that into account to look out for like what types of splicing could happen that might lead to an outcome that we weren't thinking about. And so they use a tool called a gene trap or a splice trap in order to figure out if splicing is happening to specific sequences of DNA. And from that, they build a hypothesis of how the splicing event might lead to some of those adverse events that we see with the ChAdOx vaccine.
Yeah, it's interesting because because we did look at like this early on about like what was causing VITT, vaccine-induced thrombocytopenia, and this is, and there were a couple of different kind of theories thrown out there, like that reacting to the actual virion envelope. So was there a reaction to the adenovirus, or was there something happening with the spike sequences? And this is like kind of the most solid like kind of look at this, because they also compare like the 3 different types of adenovirus vaccines, because we've got like the adenovirus 5, which was created in China. There was the ChAdOx, which ended up in my arm, and then there's the Johnson & Johnson vaccine. Yeah, and they find that they each have like different, so they each have splicing regions, and I think the interesting thing here is that I kind of expected that the vaccine manufacturers would have paid attention to this early on when they're in their like code and optimization, and this group actually got all the sequences from there, and then they tested them out, and they found that actually.
Sorry, can I just pause for a second? The 85 and the 8026, those are the Sputnik ones, right?
Yeah, they're the Sputnik ones as well.
Yeah, Sputnik ones. Yeah. Okay.
So they've been used in different things, but again, I don't think they've managed to see what the exact sequence for Sputnik was. In this paper. So this came from other science labs.
A different one.
Yeah, and they
but
tested
yeah,
that.
Sorry, you were saying they codon optimize, right? Because they want to make sure that when they inject this DNA, or when the DNA gets delivered through the adenoviral vector, like those cells are going to make spike. So they want it optimized for humans in terms of expression.
The thing that they didn't really take into account is that quite a lot of the SARS-CoV-2 it exists in the cytoplasm outside of the nucleus. So in the nucleus is where all the splicing happens. So once you have DNA, and that has to be transcribed in the nucleus, which means that suddenly RNA in the nucleus is subject to splicing, which means that what you get is, for some of these vaccines, you get a truncated version of spike protein being produced. And they tested the relative amounts of.
Yeah, because these it's never been, right, like these sequences that we're making of these sequences of mRNA, right, that exist in the infectious, right, virus, we're changing it into DNA, and that's a DNA in the nucleus, which, you know, that's not the evolutionary constraints that these things have seen. So like, because we designed it, like we have to be looking out for these things, is sort of what the paper is telling us, that like evolution didn't look out for the potential for splice variants, and like we have to look out for it, because the truncated versions that get made during a splicing event, they might be soluble in the bloodstream, right, and floating around and causing effects that like that the virus itself doesn't cause.
Yeah. And what they did then was so that's very important. And they did test that on actual cells. So they tested it on like HeLa cells and a couple of other different cell types, and they found that different cells treat them differently. I think because of the way that.
Yes.
And I think.
Well, splicing machinery is sort of one of those. That it's one of those regulatory layers, right? Like that makes sure that from the same genome we get all this different diversity in cell types in our multicellular body. So I'm not surprised, right, that it has different effects in different places. But once again, it also reiterates to me is like, it would be really nice to know mechanistically like what tissue types are being infected by by this, but I guess we have more understanding on the viral side because this is like the adenovirus vector.
Yeah, and I think they found that of the adenoviruses, the Johnson & Johnson vaccine had the least amount of splicing, and that produced the fewest. So that didn't produce as much truncations compared to say Chadox or adenovirus 5. So I think that kind of highlights another thing about this, which is that these vaccines did have this side effect, but that it's not like a death to the vaccine as a whole. That we can correct for that. Now that we know what the issue is, we can actually make that safer. And it's not something that's inevitable.
It could be that for RNA viruses in the future, right? We don't have to discount this particular vaccine platform, right, as a way to deliver just because of a potential vaccine-induced, right, side effect. Like they can be designed at the earlier in the process to eliminate that potential.
I mean, I'll go one better. This can let's say if you deliberately added a splice that had a different variant of that virus protein, and then you could end up having with just one vaccine creating multiple variants and producing multi. So there are opportunities.
Sure. Yeah.
So it's just that, but yeah, unfortunately, because they didn't seem to be taking that into account when they created these vaccines the first time around, that this has happened, and that next time around they'll account for it a bit. But I hope they'll account for it better.
Right. Yeah, it's interesting to me because sometimes I think that if you just watch the news, you might get the sense of like, oh, the mRNA vaccine, this is the future of all vaccines, and like, right, like all the eggs are in this basket, and like, think of all this great stuff. But actually, every vaccine platform has its own benefits, right, and risks in terms of using them.
Yeah.
I mean, just talking about like the fact that we didn't get that protein subunit based one until like a lot longer, like that still saddens me to this day of like, in terms of like the benefits of that vaccine platform, like there's great benefits to that one. And I feel like sometimes the media doesn't cover it much. So, we care about many different types of vaccine platforms, and we understand that like there are advantages and benefits to them. So like don't fall for the hype sometimes in terms of like what the best technology is out there.
Sometimes they're all like above a certain threshold that we'd expect for. I mean, considering that the influenza vaccine, you can, if you're allergic to eggs, it might not be good for you, or like there's lots of other things that are there, but it's there is no such thing as risk.
Yeah, there's many factors there in terms of like finding the best one, and they can all produce good immunity, but like manufacturing comes into play. Who can't and can take certain vaccines. It's important to have a selection of technologies, I think, to choose from, and also the knowledge to use them in the best way that they can be used.
Yeah, I think there's a lot of like vaccine communications that needs to happen, as well as like access. There's a lot of stuff around the vaccines beyond the science that is very hard to get through. But yeah.
All right. That brings us to the end of. We actually had a lot of SARS-CoV-2 papers this week, despite not being like a be afraid of SARS-CoV-2 sort of week. But in this last three, we have some of the other selections from the world of microbiology, starting with injection and extraction from single fungal cells.
Yeah, this is I like this one. This is it's very physical because what it's talking about is effectively just like, because there are lots of ways to introduce genes into fungi and other like microbes. There's electroporation and holes will fall, and then stuff will get in there. Or there's like using kind of injection systems or like mating systems. But this one,
conjugation, mating system, conjugation systems. I feel like that's a pretty common one. But yeah, here we're just gonna pierce things and directly inject stuff. Tiny syringes, if you will, if syringes were shaped like pyramids and on the size of a nanoscale.
This is using an atomic force microscope to like puncture the surface. It's because it reminds me a lot of like in vitro, like those in vitro fertilization, or like when you're like genetically manipulating like mammals, where you have to get an oocyte and then you have to inject them, and it's just such a nightmare.
Actually, I was going to say it's a very popular technique in Danio research, in zebrafish research, to inject morpholinos, to inject like pieces of RNA that like genetically modify those organisms. And so, I mean, scientists have been using injection, I guess, as a research technique in sort of a wide variety of contexts. But if we think about single cell injection, that's like a lot more difficult. I mean, oocytes or embryos, they're huge. They're huge. They're the biggest cells that exist. And so yeast, in this case, we're talking about yeasts and fungal hyphae, like those are on a totally different scale. And so they require an injection mechanism that is a totally different scale. It's a tiny, here's an SEM image of it, just a tiny pyramid with a little hole in the end. And it's built on this platform of an AFM cantilever. So we've maybe briefly touched about AFM before, but essentially it's like there's a lever that extends out that they can use electricity of some sort in order to apply a force on that lever, and then that tip it interacts with the surface, usually based on some like electrostatic forces, but they can also like have affinity tips and stuff like that, and it can like scan along the surface, essentially hover along the surface of a very small things to get a sense of the features, because the depth that the cantilever moves at is controlled by electronics and has feedback into electronics. But in this case, they sort of ramp up the force, and they just use that cantilever to poke into the cell wall.
Yeah, and so they tried it with like a lot of human cells, like HeLa cells, and it's very effective. But with fungal cells, it's different because they got a cell wall, which means that sometimes when you poke them, they just slip out on either side, which is just
they're armored.
Yeah, which is just like very like annoying, I can imagine. So they have to like redesign the surface to make sure that those cells stay in place, and then they can do either inject or take stuff out, which is again quite a fun. So firstly, like one thing they do is they inject fluorophores, so things that make them glow in the dark, and then see whether the creatures stay alive afterwards.
Yeah, and they poke things, they do the injections, and then they just watch over time what happens to those cells that they injected in. And yeah, it seems like these cells are surviving. And then, as always, this is a technology platform that they're talking about, but they want to show us, some useful takeaway, right? Some useful data to take away with this technique. And what they do is they show that it's been known in the field that fungi respond to being fed on by expressing certain proteins, but by nematodes. But it's unclear whether or not that is produced by like the whatever molecules are in the nematode's mouth is like what the fungi are recognizing and sort of upregulating the expression of certain things using, or is it just that action of extracting out all the goodies inside of the cytoplasm that causes this upregulation of genes? And so you can imagine with a tip, there's no, saliva. I mean, I don't think they even have saliva, but there's no like mouth molecules to contaminate the bite wound, if you will. And they're able to show that there's an upregulation. Yeah, it's this one. Yeah. There's an upregulation of a certain gene just by piercing and sucking a little bit of liquid out.
Yeah, it's like that image of like, because the nematode worms have these little fangs that they dig into the fungus and suck stuff up, and they were looking at their thing going, well, actually, our kind of thing looks like fangs as well. Let's just do the same thing and see what happens.
Yeah, but of course, there's many other applications that they don't go in, like they discuss, but they don't actually directly show, right? Like this idea you can inject something, a very specific molecule that makes a genetic change in that area or changes the metabolism of that specific cellular area. You can sample, right, the cytoplasm at a specific moment, or, I mean, I think importantly in the fungal system, they have these huge, right, like hyphal networks. Like you can choose a specific zone of that and have this resolution of understanding what metabolites are at very specific points, instead of, again, just grinding it all up and taking the average.
Yeah, completely. So this is a really interesting technology because it also seems like deceptively simple. Like just why don't you just poke a hole and why don't you just inject the fungus back? But in order to do that, we had to get all these other technologies ready in the first place.
Totally. It's a very small scale, and they're very tough cells. So, all right. Up next is toward a glycyl radical enzyme containing synthetic bacterial microcompartment to produce pyruvate from formate and acetate. So actually, I think in this last deep dive vote, we were going to talk about microcompartments potentially, nanocompartments, right? Essentially, the discovery here, or the knowledge is that bacteria have complicated cytoplasms, even insofar as they have specific protein-based compartments that isolates the enzymatic reactions from the rest of the cytosol. And with that knowledge, engineers and sort of technology folks are saying, well, let's see, can we create our own that do specific biochemical reactions that humans care about? And I think here they're thinking about a type of harnessing. It's sort of known that you can engineer different metabolisms to create formate as like the starting point. And so it'd be really great to have subsequent reactions that then take that formate and prepare it to become a whole bunch of other things. And sometimes those enzymes require like a very specific microenvironment. So that's where bacterial microcompartments can come in. This is a great picture that you brought up of like how the wild type microcompartment looks in A, and then they were looking at removing key pieces of the genes, and it turns out those compartments still assemble, and they can even assemble with holes in them so that it can allow for the diffusion of different molecules in and out of those particular microcompartments. Yeah, they call it a wiffle ball. The minimal wiffle ball is the way of thinking about these things. And yeah, and so on the inside of these compartments, you'd have this privileged zone, right, where certain types of metabolism can take place, and you won't have to worry about interference from other enzymes, or even like potentially if the size of the holes can be restricted, or maybe in the future gatekeepers of those holes, right, can be created. You'd be able to really control the biochemistry that's going on inside of these.
Yeah, they're really intriguing little nanomachines that are. Yeah, I love the fact this paper goes into the design of them and how they went from, say, nature, where they looked into nature and they found, like, I think they went to an organism where, like, let me double check, but
it was a Myxococcus, I
yeah.
think, which is like one of these like social amoeba styles. I mean, I have the diversity, right? We always talk about this. There's huge diversity within the bacterial world. Yeah, you never know where you're going to find an interesting structure that you might want to use or co-opt into a different process.
Yeah, and also the process they use for creating formate. I think we talked about this in the previous show, where they essentially took a process where they use formate to create pyruvate, and then they create conditions to make that reaction run backwards, which again is just quite a cool thing. So this is got quite a lot of like interesting ideas, and the thing that excites me is like what you can build from this. I mean, just going back to the original paper, the previous paper, where what can we inject into fungi? Well, we can inject wiffle balls into them to do some kind of metabolic stuff, or like if you want to get really out there, like, okay, well, how can we could use these to do stuff for human metabolism as well, potentially, because this own like kind of segregated from the rest of the system, so they are like closed.
We have to ask the question, you know, I mean, there's so many follow-up questions that come from that, but it's a really cool idea. Like, what's the immunogenicity of these objects, right? Like, can we make them less immunogenic, right? I mean, that would be amazing, right? Then these are like self-assembled, right? Like objects. So I think that's the other thing to talk about here is that I think people think that, you know, a lot of evolution, like, is taking things that already exist and modifying them for the own individual purposes of the organism that has them. People look at these microcompartments and they say, this is like a lot like a viral capsid. And perhaps like that's where the evolution has come from. So in that manner, they self-assemble. So merely expressing these proteins, they're sort of in a certain type of shape where they just kind of like all interlock in a certain way. And so the researchers, for one of those interlocking pieces, they engineer like a tag, essentially, that allows them to associate a number of enzymes with it. And so you can see in this picture, you can clearly see that one of those subunits has the enzymes attached to it. And so when the self-assembly happens, those enzymes end up on the inside of these microcompartments.
Yeah, and that means you can actually, like, it's a great way to like make metabolism more portable. So you can transport some metabolism between different cells, and because of the way microcompartments are designed, you can have it still regulated. So those compartments have things slightly different set up. So it's there's a lot of
like the stoichiometry is well controlled, right, inside of the microcompartment. Potentially.
Yeah, I guess this is one of the things where I'm quite interested to see where our imagination takes us with this technology, because.
Absolutely. All right, and final thing for today we have is structural basis for mismatch surveillance by CRISPR-Cas9.
Yeah, this is so CRISPR-Cas9. It's a system that everyone's talking about for gene editing,
and we'll be talking about forevermore, I think.
Yeah, but one of the issues with it is it isn't exactly 100% great. So there is a lot of like mismatch targeting. So sometimes it can cleave stuff that you don't want it to cleave. Yeah. And this paper
I just want to say, like, So, and that's like borne out, if you really dive into sort of gene editing literature, they'll always say like, what are the off-target effects, right? And if you make like a specific genetic change using CRISPR, then they want to see a really important piece of data is to then see that whole genome sequence of the thing that you changed, just to scan over it and make sure that like there aren't any other changes.
Yeah, I think that if ever, that's really important, because I mean for like, I know that for gene therapies, it's been a very hard road because of off-target effects that can cause really bad side effects. So this is quite an important thing. And the way they answered this question was they actually looked at how the CRISPR-Cas9 protein actually works by doing a bunch of one of my favorite techniques, which is with 3D cryo-electron tomography, where they look at the structure of it and then look at it in motion effectively. So watching it, well,
they sort of see like hundreds, thousands, millions. I'm not sure, but like they take pictures of tons of these CRISPR molecules bound to a piece of DNA, and you're bound to find in all of those still images, right? Like intermediate steps of motion that are going on.
Completely. And so what they look at like each different component and looking at how, like, so you got your CRISPR, which is a DNA molecule that is used to match to the DNA strand that the CRISPR is looking for. And it has what they effectively found is that there's that there are certain like proteins that help CRISPR to make a positive match even when the DNA doesn't quite match. There's this. So they're very different sections of the CRISPR protein, and I think there's I think the one that's important here is this thing called rhesus, which almost like acts as a finger to like press the. So usually what we'd hope for is when DNA like kind of matches together, it kind of like comes together in like a zipper kind of fashion, but when like there's a mismatch, and that would usually cause the entire thing to stop. But what happens is this like protein comes in and it pushes mismatches together, so it still causes the gene to the CRISPR to roll and to cut even when it doesn't entirely match. Which I can imagine is quite useful for when in CRISPR's native environment, when it's there to fight viruses that mutate a lot. I can imagine that being like quite good for survival, because that means that if the virus mutates away from something, then the CRISPR can still attack that same virus. But when you try and look for what we wanted to use it for, we want it to be specific, that kind of extra thing, that extra finger that presses the DNA together is less useful to us. So what they did here was they took that finger and they stiffened it and made it so it didn't work as well, which meant that the CRISPR will only like work when it's got a solid target.
Wait, I guess not stiffen the finger. They probably like loosen the finger, right?
Loosened it. They mutated it in a way that wouldn't cause that same effect, and because of that they made CRISPR a lot more like effective.
Yeah, very cool. Like this is an interesting paper in that it's taking information from the structural domain, right, and like translating it directly into engineering, designing, right, like protein engineering type thing. Like we often see structural data in informing us how something works, but then to see it feed back into then how to then change how something works, that's pretty cool.
Yeah, I think that's basically what I have to say about that paper.
Awesome. Well, join us next week for more cool microbiology news.
Yeah, don't forget to like, subscribe, and do what else. And thank you for making it this far. We want to remind everyone that while we're very enthusiastic about microbiology and somewhat qualified, it's possible we didn't get everything right. Science is about thinking critically and asking the right questions. So if you have any questions or corrections, please let us know in the comments.
Yep, I totally agree. You can reach out to us over Twitter at microtwjc. We both believe that peer review is a process that we can all participate in. It's great to have more people reading papers. And so if you've had a good time listening to us or ramble on about microbiology today, if you have something to add or found unclear, just let us know in the comments.
Yeah, it's been a pleasure chatting with you, Danny.
Same here, Fuzz.
Tune in next week for more microbiology content. Goodbye.
Bye-bye.